Hello students, welcome to my online class of today, which is something about poriferans. You must have studied about Cycon or Scypha, okay, which is a representative of the phylum Porifera. After knowing its morphology and body wall, you are expected to know yet one more aspect of Cycon, that is sexual reproduction in Cycon. On the slide, you can see two names, Cycon oblique Skypha. What do you understand by that? See, both of them are zoological names, ICZN names only, referring to the same poriferan. Cycon is the old name and Skypha is the new name. So, whenever a new nomenclature is given to the animal, you have to use the new one, not the old one. Okay? So, correctly speaking, you should use Skypha, not the Cycon. Okay? So, today we are going to learn about sexual reproduction in Scypha. Okay? Let me introduce myself. I am Dr. MEERA B K, Associate Professor and Head, Department of Zoology, Maharani Science College for Women, Palace Road, Bangalore 1. On the slide, you can see two larvae, okay, which are the larval stages met with when you are studying the sexual reproduction in Scypha. Okay? One is amphiblastula larva and the other one is parenchyma larva. Okay? Now let us begin our class with this background information. Now, some of the sponges, no doubt they are unisexual, in the sense you find separate male of the species and female of the species. Okay? But most of the sponges are monoecious or bisexual. Monoecious or bisexual is opposite of unisexual. That means both the gonads, male and female gonads, are present in one and the same animal. Then the condition is referred to as monoecious, mono means one, or bisexual, in the sense in the same individual you find both the gonads. Okay? So, most of the sponges are monoecious or bisexual. Very rarely you do find few unisexual sponges too. Although they are bisexual, that is hermaphrodite. Hermaphrodite is the term used to indicate monoecious species or bisexual species. There, self-fertilization is avoided and cross-fertilization occurs as a rule. Self-fertilization is the sperm produced by the same individual fertilizing the ova produced by the same individual. It should not happen. Okay? So, in sponges that self-fertilization is avoided, it is not good for perpetuation and continuity of the characters. So, cross-fertilization is a rule. Okay? How self-fertilization is avoided? The sperm and ova, are formed at different times. Okay? They are formed during different times, so that when sperm is mature enough to fertilize, ova of the same animal is immature enough and vice versa. So, though they are hermaphrodite or bisexual or monoecious, cross-fertilization is the rule. Self-fertilization is avoided. So, cross-fertilization is possible because the production of sperms and ova, the timing differs. The sperm and ova basically they are derived from undifferentiated amoebocytes called archaeocytes. About different types of archaeocytes you must have learnt in your previous class. So the gametes, that is male gamete is sperm and female gamete is ovum, they are derived from undifferentiated amoebocytes called archaeocytes. One more question, please note down students. Then the sperm and ova are also known to be derived from choanocytes. Apart from archaeocytes, there is another source, okay, there is another source of the gametes, that is choanocytes, which form the inner lining of the spongocoel, right, which later undergoes gametogenesis to form sperm and ova. So there are two sources for the production of gametes. One is archaeocytes and the other one is choanocytes. Okay, choanocytes and pinacocytes, you must have learnt about them when you studied body wall of cycon, and archaeocytes also you must have learnt. So right now you just know that the gametes have double source. One, they are formed from undifferentiated archaeocytes, and second source is choanocytes. So gametogenesis takes place, resulting in the formation of sperms and ova. Okay, then now sponges exhibit protandry and protogyny. You have to know about these two terms. Okay, so what is protandry? Andry means something referring to the males. Gyne means something referring to the females. Pro means first. Okay, protandry means production of sperms takes place first. Okay, and later the same individual will start producing ova. So if sperms are produced first, followed by the formation of ova, then the situation is known as protandry. On the other hand, if the ova are produced first and sperms later, the condition is known as protogyny. So these are the two terms which you should know, protandry and protogyny, and the sponges exhibit either protandry or protogyny, since both the gametes are not formed synchronously, that is at the same time. One can easily avoid self-fertilization, so thereby cross-fertilization becomes a rule. Okay, so be aware of two terms, protandry and protogyny. Okay, then both of them facilitate cross-fertilization, as I was telling you. Now let us talk about spermatogenesis. You know that gametogenesis is the formation of gametes. Formation of male gametes is known as spermatogenesis, while the formation of ova is known as oogenesis. Now you have to learn about these two processes in Scypha. Okay, so the sperm mother cell or a spermatogonium, as I was telling you, is nothing but an enlarged archaeocyte. Okay, it is an enlarged archaeocyte, and this is surrounded by one or more flattened cover cells to form spermatocyst. Okay, to form spermatocyst. So to begin with, a sperm mother cell or a spermatogonium is identified, which is nothing but an enlarged archaeocyte, and later this is covered by flattened cover cells to form spermatocyst. Spermatocyst is formed. Then these cover cells are derived from amoebocytes. Please note, to be sperm is derived from archaeocyte, whereas the nurse cells or the cover cells are derived from amoebocytes. Please note the difference. Then the spermatogonia undergo 2 to 3 maturation divisions to form spermatocytes, and these spermatocytes later give rise to spermatozoa or sperms, functional sperms, spermatozoon singular, spermatozoa plural. A mature spermatozoon consists of a round nucleated head and a tail, and the lashing movement of this tail help the spermatozoon to reach the other sponges. You know that self-fertilization doesn't take place, cross-fertilization is the rule. Therefore, the sperm has to escape from its parent and parent animal, and then it should reach another sponge to fertilize the ovum, right? So, the lashing movement of the tail helps in this journey, all right. This is all about spermatogenesis, okay. So, let us learn about oogenesis. The egg mother cell or an oocyte, it is also derived from large archaeocytes which have distinct nucleus, okay. Egg mother cell or an oocyte is derived from large archaeocytes which have distinct nucleus. Sometimes the oocytes also arise from choanocytes, as I was telling you, and the oocyte moves like an amoebocyte engulfing the other cells, okay. Then these engulfed cells act as nursing cells or nurse cells for the developing oocyte. The fully grown oocyte undergoes 2 maturation division, as you see in spermatogenesis, to form ovum. The ovum lies in the wall of the radial canal or spongocele, and now it is ready to be fertilized by the sperm produced by the other sponge of the same species, okay. So, after learning about spermatogenesis and oogenesis, you should learn about fertilization. Sperms are released out from sponge through the outgoing water currents from the osculum. By now, I suppose you know what is an osculum and what is an ostium. The large opening in the anterior end is the osculum, and where a number of pores all over the body are the ostia, ostia plural and ostium singular. So, sperms are released out from sponge through the outgoing water from the osculum, and these sperms which are released make their way into another sponge through incoming water by ostia, okay. Then choanocytes act as nurse cells and transport the sperm to the ova, and they lie right now in the flagellated choanoderm, okay. And the fertilization is internal and cross type, okay. Now you have learnt about spermatogenesis, oogenesis and fertilization. Now you have to understand about the process of development. So, early development takes place within the maternal sponge. Maternal sponge is the one whose ovum is being fertilized by the sperm produced by the another sponge of the same species, and this results in the formation of larval stages. Please note the development is indirect, involving 2 larval stages, okay, what I had shown you on the first slide, right. And if there are no larval stages, then you say development is direct, but here development is indirect. There is production of 2 larvae in the life cycle. The larval stages bear the flagella which help them to escape out from the maternal sponge body. The larva thus escaped gets attached to a suitable substratum, metamorphose and grow into an adult sponge. Please note, poriferans are sedentary, right? Adult ones are sedentary, whereas the larvae are motile. Okay, they are motile because of the presence of flagella. And now all the time I was telling you that you find two larval stages in the life history of sponges. So sponges have two types of larvae. Now let us see what are the two types. One is amphiblastular larva. It looks like this. It is a hollow oval larval stage, characteristic of calcareous sponges, that includes Scypha also. Okay, it's a hollow oval larval stage, right? And you can see from the diagram, anterior half of amphiblastula bears flagella, okay, while the posterior half is free from flagella, right? This is about amphiblastula larva. Apart from amphiblastula larva, you find one more type of larva called parenchyma larva, and it is solid oval or flattened larval stage. Please note, amphiblastula is hollow, parenchyma is solid. Both are oval, parenchyma can be flattened also, right? And that is the amphiblastula is the characteristic of calcareous sponges, and this is also characteristic of calcareous sponges, especially the order Hexactinellida and Demospongia. And the entire larva is covered by flagella here, whereas in amphiblastula only anterior half of the larva has flagella, whereas in parenchyma whole larva is covered by flagella. Okay, this is the difference between amphiblastula larva and parenchyma larva. Okay, then, so with the help of external flagella, the motile larvae escape from the parental body, swim for few hours to many days, and then they settle down, become attached to some solid object or the substratum, undergo metamorphosis and grow into an adult. By now you must be knowing what is metamorphosis. Metamorphosis is a process where the larva loses its larval characters and acquires adult characters, so that at the end of metamorphosis the individual resembles more of an adult, right, but for poor development of reproductive structures. Students, with this I have completed the sexual reproduction in Skypha, okay, and few questions to answer for you is question number 1. Discuss the larvae included in the development of sponges. That can be asked for 5 marks, wherein you will discuss both the larvae, diagram and the description. Or same thing can be asked in the other way, describe sexual reproduction in Cycon or Scypha. They may use Cycon also, you should know, but Scypha is the correct newer nomenclature. So the second question can be asked for 10 marks, first question for 5 marks. So with this I have completed my talk on sexual reproduction in Cycon or Scypha. Okay. Now, students, thank you for your patient listening. Any questions you do can contact me. Till then, stay blessed.